Okay, this is chapter 14. It's called nervous tissue. There will be a series. This will be a series of 3 separate videos, but they'll be long, so you might want to take breaks. And the first one deals with the tissue, neurons, neuroglial cells, characteristics like that. The second one deals with the brain and the cranial nerves. And the last one deals with the spinal cord and spinal nerves. Now I'll take that back, there's 4. And the last one is the autonomic nervous system. Okay, so the nervous system is obviously the most complex system in the body. It's also kind of dry. So you need to do something to keep you focused because it's really, really important stuff. So here we go. Nervous tissue. You know, we always begin. Man, I got a mouse here. This is the second time I've had to record this. The first time it didn't seem to work. Okay, there's the arrow. Okay, so first thing we always do is we always do an outline. Yeah. And we talk about in the outline what we're going to be discussing in detail. Okay, how is the nervous system organized? The cytology of the nervous system. Remember when we did tissue and we had categories? So for an example, epithelium, there was about a dozen subcategories. Nervous system, there's 1. Neuron, that is the nerve cell. There's helper cells, and those are called neuroglial cells, sometimes referred to as glial cells. You've heard of myelin, and we're going to find out now what glial cell actually lays down the myelin and what the function of myelin is. Okay, not all cells are myelinated, and only the axons of the cells that are myelinated. We've talked about a neuron not able to undergo mitosis, and that's true. Once you have it for as long as you are alive, or it doesn't get damaged too bad. What they find is in the peripheral nervous system, part of the axon is able to regenerate. If it isn't too far, now go back to where the cell body joined the axon. Remember, it was a place called an axon hillock. If the damage occurs too close to the hillock, the cell dies. If it's far enough away towards the axon knob or the telodendria, it can regenerate that. So we're going to talk a little bit about that. What are nerves? I'm looking. See this wire? If I asked you what was in this wire, you said there's more copper wires inside, and you'd be right on. The copper wires represent a neuron. All those neurons wrapped together make a nerve in the peripheral nervous system. If this same thing is in the central nervous system, which is the spinal cord and the brain, it's called a tract. Same thing, tract in the central nervous system, nerve in the peripheral nervous system. Synapses. Okay, remember when we did muscles, and we talked about how a muscle contracts, and then we talked about an axon knob and the sarcolemma and the synaptic cleft. Remember that? Okay, we'll touch on that again, only this is not going to be a neuromuscular junction. This is going to be a neuro-neuro junction. Good? I'll see if I can make my mug a little bit bigger. Okay, it looks like it might work. Hold on. There you go. There you go. You don't know this, but I got my pajamas on. Yep. Okay, synapses. Now, this says neural integration, neuronal pools. Neuronal pools. Sometimes several neurons feed into one neuron. So let's say my fingers are neurons and they feed into a single neuron. That's called integration. Going back the other way, one can feed into a bunch of them, which is usually what happens in a motor neuron, and that's called integration separation. It's not the right word, but it's coming up. Okay. Anyway, not just simple one-on-one. Development of the nervous system we will not discuss. Even though it's in your textbook, you're welcome to look at it. That's embryology. Okay, organization of the nervous system can be divided into two categories. Okay, it's not the central nervous system, peripheral nervous system. It's structural and functional. Okay, structural is the type of neuron, which is going to be unipolar, bipolar, or multipolar. And functional is it sensory or is it motor? Get it? Okay. Let's take a look at those from the structural part. There are two subdivisions of the nervous system. Central nervous system, which consists of the brain and spinal cord. They abbreviate it CNS, central nervous system, and peripheral nervous system, cranial nerves, spinal nerves, and ganglia. Think of it this way. Picture your spinal cord as a tree trunk, and the leaves and stuff in a cluster on the top of it is your brain. So you have the spinal cord and brain. That's the central nervous system. Now, anything that comes out of that is called the peripheral nervous system. And there's two places that it comes out at. It comes out at the top of the tree, out of the brain. Okay, and those are called cranial nerves. And it comes out of the spinal cord, and those are called spinal nerves. Got it? Okay, picture really doesn't mean anything. Just showing you, here's what I was talking about. Okay, that would be the trunk of the tree, which is the spinal cord. Here's the canopy of the tree, the leaves and stuff, that would be the brain. And you see what these gold things are. These are cranial nerves that come out and feed part of the body. And these are all spinal nerves that come out and feed the rest of the body. Anytime you come away from the central nervous system, that is the peripheral nervous system. Okay, now sensory and motor. It says both the central nervous system and peripheral nervous system have three subcategories. What their function is to collect information. Obviously, you know that's going to be the sensory side. Okay, and we do those with receptors, photoreceptors for the eye, chemoreceptors for the nose and the mouth, touch receptors. We already did Meissner's corpuscle and Pacinian corpuscle. Okay, so we sense the information and collect it. Okay, and then we send it up to the processing center, which is the brain. Once it's processed, we may or may not do anything with it. If we do something with it, that part is called the motor part, which is what responds. Okay, so let's see if I got this going here. Yep. So we collect information, sensory, we process it, brain, and we do something about it, which is motor. Good? Functional divisions of the nervous system. Okay, here we go. So this is straightforward. Sensory. Okay, we have two functions. You can kind of figure them out. Here's to you. I gave these up for a day. Don't do it. It's not worth the 240 calories. Okay. Hey, any of you guys gaining weight like me? Boy, it's pretty easy being around the house, huh? Let's hope this doesn't last any longer than necessary. By the way, you guys saw that temperature is supposed to be up in the 80s on Wednesday. Gonna be hard to stay in the house with that, huh? Just bear with it. Bear with it. Think how nice it's going to be when we can actually get out. Anyway, get back to the system. We have two systems, subdivisions of the functional part, It's sensory or it's motor. Okay, it's sensory or it's motor. Okay, this chart, even though it's a little boring, I would think that you like the ones that I do that have the boxes and the clouds and the different colors. Because you can see, here's a good example of an informational figure, a chart, has everything you need to know, but you look at it and it's boring. Whereas if you took the nervous system and made it let's say a black box, and then you made the sensory nervous system blue and this blue over here, put a little cloud around it, catches your attention. Okay, if you don't want to do it, that's fine. But this pretty well tells how the nervous system is broken down functionally. Okay, you got the nervous system divided into two parts, sensory and motor. We just did that. Okay, and it talks a little bit about what sensory does and motor does, but that's kind of a no-brainer. Okay, under sensory, we have somatic, which means, remember, somatic always means skeletal muscle. Okay, good. What the heck was that? Okay, skeletal muscle. Visceral applies to, here we go. Visceral applies to smooth muscle, cardiac. So we have skeletal muscle, smooth muscle. This is not working. Let me go back to this. This is smooth muscle, cardiac. This is skeletal muscle. Remember, the nervous system is telling it to contract. On the motor side, we have the same thing. See, somatic motor, sensory motor. But instead of visceral for the motor, it's called autonomic, because it's going to have two subdivisions. But it's still mostly going to visceral. There is some somatic stuff going on. Sensory, look at this, another word. Sensory, also called afferent nervous system. Afferent, A-F-F, same thing as sensory. We're going to have a different one for motor here coming up. Okay, what does it do? Now this is a repeat. It receives sensory information collected from your receptors, your photoreceptors, your chemoreceptors, your mechanoreceptors, okay? So they're just repeating this. But the new term here I put in green, sensory and afferent are the same thing. Okay, subdivided into two groups. Afferent, sensory. You already know it. What we're doing is we're talking about that chart I just did. So remember sensory? It was on the left-hand side. What did it branch into? Somatic, which went to skeletal muscle, and visceral went to smooth and cardiac. Somatic and visceral. Do you see it? Okay, so all we're doing with these slides is we're going over that chart. Okay, good. Motor. This is the next subcategory. So we're on the other side of that chart. Also called efferent. Sensory, afferent, motor, efferent. Good. It says it sends impulses from the CNS, which is the brain and spinal cord, to the muscles and glands. Two subdivisions. Go back to that chart. What did you see? You saw somatic. So, it goes to the skeletal muscle. And what was the other one I said was pretty much like visceral? Picture it. Okay, it was autonomic. And remember I said there's two subdivisions there. Somatic motor and autonomic motor. Good, good. You see where it's down here where it says regulate smooth cardiac muscles, glands. Okay, there is going to be some skeletal stuff, but not much. Okay, so we'll just keep it at that. Neurons. Okay, now remember in the outline we said what is the nervous system and what is its subcategories and that kind of stuff. Now we're going to go into the neurons. We're doing cytology. It's pretty simple because there's only two. there is the neuron, and associated with it is the neuroglial cells, sometimes called glial cells. Cell of the nervous system. Okay, what does it do? Big deal. Now, remember when we were in lab, if you can remember back that far, remember I had two different types of models of neurons. One was on a wooden board standing up, and the board was brown, and it had kind of a turquoise cell in it, and you guys had broken off the dendrites, but it had a nice axon, telodendria, and it was attached to a strip of red plastic. That red plastic represented muscle, and that was skeletal muscle. So obviously that was a motor neuron, and that was a somatic motor neuron, okay? The other one was, I had two of them. One was on a green plastic table, and it had the cell body and the dendrites and some little tiny kind of flesh-colored bulbs attached to the dendrites, which we knew were synaptic knobs of another neuron. And then next to it was the axon. You remember that? Okay, that's what we're talking about right now. These neurons. Those were neurons. Okay, characteristics of a neuron. High metabolic rate. Well, yeah. Without getting into the physiology, if you guys could decide to take A&P from me, I'll talk about why there's a high metabolic rate. Right now, this is just a fact you need to know. It has a high metabolic rate, okay? Extreme longevity. I put an asterisk by this. This comes right out of your textbook, and it says extreme longevity. You can't get any more extreme than your entire life. That's why I put the asterisk. Kind of like a dumb statement, but I put it so you'd recognize it. Came out of your book. Non-mitotic. It doesn't undergo mitosis. If you lose it, you lose it forever. That kind of goes with extreme, doesn't it? Okay, structure of a neuron. There's three main parts. You're going to nail this one. It's simple. Axon, dendrite, cell body. Good? Okay, here we go. We're going to list it. Cell body. That's where all the cytoplasm is, where the nucleus is, the endoplasmic reticulum, where the Golgi is, et cetera, just like any other cell. If you picture that cell, that prototypical cell we had in lab, what you do is on opposite ends, you take and hold on to the phospholipid bilayer and you stretch it like you would a piece of taffy. You know what taffy is? You don't know what taffy is. Let's say you stretch a piece of clay and you stretch it and stretch it and stretch it and stretch it just before it breaks. Those long little filaments that you made by stretching it out, that's an axon on one end, dendrite on the other, with a big mass in the middle, which is the cell body. Got it? So the next one is dendrites, of course, and axons. And the way the impulse, that means the electricity travels, it always travels from the dendrite to the axon through the cell body. So it's picked up by the dendrites, goes through the cell body to an axon. Always, always, always. Got it? Okay, here we go. Now, you saw this guy because we did it on the first lab exam. Okay, but here's kind of like an illustration. Here's what it would look like under a microscope that you have actually seen, because I asked you what those dots were on the lab exam. And those dots are dendrites. I mean, are neuroglial cells, huh? And I think that on the lab exam, this was blue. Okay, but it doesn't, just meant that was the stain. Okay, this is all extracellular. And there's your dendrites, and here's your axon. And your axon hillock means where does it start? Is about right. Where'd you go? Where'd you go? About right in here. Okay. Here's this is the nucleus, and inside the nucleolus, and this is all the cytoplasm. And we know that inside the cytoplasm, we have mitochondria and Golgi and all that kind of stuff. We just can't see it here. And this one's myelinated. This one's showing you the myelin sheath. And those little gaps, remember, what were those called? What were those called? Here, I'll take a hit while you guys are figuring it out. nasty stuff. Okay, those are called nodes of Ranvier. If you say nodes of Ranvier, you can probably spell it. Good. And remember down here, these were telodendria. And at the end of those was ET's finger, axon knob. Okay, good. These are all dendrites. Good, good, good. Okay, working some more with cytology. It says there's two distinct types of cells within the nervous system. Okay, think. What's the answer? Come on, what's the answer? Okay, should be neuron and neuroglial cell. Okay, the nerve cell is a neuron, but the helper cell is the glial cell, the neuroglial cell. Okay, neurons, nerve cells, and it says electrically, they're excitable cells that initiate, transmit, and receive nerve impulses. Okay, we know it as electricity, depolarization, repolarization. That's all what these guys do. Okay, and then we have glial cells, properly called neuroglial cells, but glial cells are the support cells. That's the big thing. They don't do anything other than keep the neuron healthy. Okay, cell body, nucleus is, I just put what's in the cell body. Okay, that's going to be all those organelles. But I put some of them so you realize, I didn't go into the big list of them, but I put some of them so you realize a neuron is still a cell. It still has a nucleus. It still has ribosomes. Still has all that same stuff, okay? So I put nucleus, nucleolus, mitochondria. Okay, don't worry about this part. But free ribosomes, the Nissl bodies, don't worry about that, but free ribosomes and which are on, are out in the cytoplasm, and then there's rough endoplasmic reticulum that has fixed ribosomes on them. I just gave you examples of some of them, okay? Same as any other cell. Dendrites. They extend from the cell body. Okay, now. Dendrites carry them to, yeah. Okay, that's a true statement. They receive nerve impulses, that's electricity, and they carry them to the cell body. We're talking about direction, okay? Okay. Axons carry, this is a true statement. Axons transmit nerve impulses, in other words, electricity, away from the cell body. So it comes into the cell body by a dendrite, gets carried away by an axon. Okay, go back to that picture. Don't worry about that statement. It just says it may or may not have an axon, but I don't want you to worry about that. Okay, this says, where does the axon begin and the cell body end? It's called the hillock. Then I showed you on that drawing about where that hillock is. Okay, now, the axons seem like they get all the attention because we have myelinated ones and non-myelinated ones. We have some that split. Let me do that again. We have some that split, axon split, so there's more than one axon. And we know at the end of axons, we can get telodendria. Telodendria, yes, with the axon knobs on each one. So axon collaterals are the branches. So when it branches, it's called a collateral. Telodendria are branches off the axon that lead to the axon knob or synaptic knob. And synaptic knobs sometimes are called axon knobs. Okay, we learned this because I drew this out for you when we did the neuromuscular junction. Okay, parts of a neuron. Just a minute here. I'm going to pause this. I'll be right back. Okay, we're back. Talk about parts of a neuron. There's a table. This slide shows the table, tells you what a neuron is, cell body. It has all those little parts we talked about. If you want them summarized, there you go. Okay. There's, according to structure, there's 3 types of neurons. Unipolar, bipolar, like your friends, and multipolar. Multipolar is the one we looked at when we were looking at tissues under the scope. Just says there's 3 types. Unipolar, looks like a upside down T. Bipolar, and I'm going to show you pictures of these, and multipolar. Got it? When we looked at them in the lab was multipolar. Okay, there we go. See the upside down T? See upside down T? This is unipolar. Let me get this back here. This is unipolar. We're going to see this one in the dorsal root ganglion when we do the spinal cord. Okay? This is bipolar. Notice long dendrite. See all this? Short dendrites. Long dendrite. This one, short dendrite. Axon starts here and goes into the cell body and then out again. Here, the dendrite goes into the cell body, but it's elongated. And this is the multipolar one, the one that you're most familiar with. It's in the skeletal muscle. See these are all dendrites feeding in to one cell body and then one axon. Okay, neuron classification by function. This is the 3rd time we've seen this. Function. You're either going to be, as a neuron, you're either going to be motor, you're going to be sensory, or you're going to be the one that connects it in between, which we're going to see as a new name that you haven't seen yet, which is called association. Okay, so here we see sensory, also called afferent, motor, also called efferent, and interneurons. Interneurons, association, 2 names. So put interneurons, and then let me find the arrow, right here, I want you to put a parenthesis like you see here and put association, 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 okay? Okay, this is a good slide. We're gonna see this thing pop up a couple of times, but what this is showing you is sensory comes in the backside of a spinal cord. This is the spinal cord, this is the anterior, this is the posterior, and there's a swelling right here that only sensory has, only the posterior, and it's called posterior root ganglion. You see the unipolar neuron, okay? And there's a bunch of them. So if you touch your finger and you feel that, it comes in the backside of your spinal cord, and then from here, it goes up to the brain. This is actually a reflex. A reflex. And that's one of the functions of a spinal cord. But what they tried to show you is this part here is the interneuron. So what it does, it comes into sensory and then it goes up to the brain. Then it comes back by an interneuron and then out of motor and has a muscle move. Do you see? So what they're trying to do in this photo is show you all 3 in one picture. Sensory, look at the arrow, coming in. Here's the cell bodies. Synapses right there. This is a neurosynapse to an interneuron. And then it goes to synapses again to a motor neuron and then out, and then it depolarizes out here. We're going to see this is in a reflex. This is called a multipolar reflex, but we'll get to that later. Anyway, there's the 3 neurons. That's what that picture's to show you. Here it's in here you see it. Now, notice it says structural, unipolar, bipolar, multipolar, functional, sensory, motor, or interneuron. What they did on this slide is they put everything together on a chart for you. Good? Glial cells. also called neuroglial cells. I like the proper name, neuroglial cells, but you'll see it shortened. I guess people didn't either know how to spell neuro or just don't want to put that all in there, but glial cells. Okay, and we're going to find out that there's 6 of them. There's 6 glial cells. Okay, 4 in the central nervous system, 2 in the peripheral nervous system. Anyway, I'm going to show you a slide on that. Here we go. Also called neuroglial cells, found both in the CNS, PNS, capable of mitosis. That's nothing new. We've already talked about that. Glial cells can undergo mitosis. Neurons cannot. What do? Protect, nourish. In a lecture format, what I would say right now, and I'm going to do it for you now, is picture that you have, at birth, you're given a car, and they say that car has got to last you for your whole life. Otherwise, car, neuron. And you need tires, you need the oil changed, you got to put in fuel. It might get wrecked and you have to have it repaired. Yes? Your mechanics that would take care of your car are your glial cells. And each one is specialized. If you needed to have engine work, you wouldn't go to a tire shop. So that's why we have the different types of glial cells. Do you understand? Okay? Got it? Okay. Glial cells. And if your car gets too messed up and it can't be repaired, you're done and you're walking, which means you don't have a neuron anymore. Okay? Okay, obviously they're more numerous. You saw it under the microscope. Those little dots were everywhere. Those are glial cells. Okay, when we hear somebody has a brain tumor, normally, I'm saying better than 95% of the time, it's the glial cells that have undergone neoplasia. The glial cells have lost the ability to stop mitosis. So uncontrolled mitosis, we know, is neoplasia. And that results in a tumor. So usually brain tumors or nervous system tumors are caused from the glial cells, and we call them, collectively, we call them glioma. It isn't the only way it can happen, but since they have the ability to undergo mitosis, those are the ones that are going to start mitosis and never stop. Anyway, a glioma is neoplasia of the glial cells, okay? Picture, don't even look at it, it means nothing. They try to do a prototypical neurons with glial cells and show all the different types. It's too confusing, don't even worry about it. Okay, let's go into the glial cells. There are 4 of them in the CNS. 4 types of glial cells in the CNS. Here they are listed. It's a no-brainer. You're going to see this on the exam because it's easy to make it into a multiple choice, which is not. Okay, astrocytes, epidermal cells, microglial cells, and oligodendrocytes. So what I'm going to do is I'm going to take each one of these and I'm going to talk about characteristics of them. And you're going to need to know that because in an exam situation, what I'll do is I'll give you a scenario and then ask you which glial cell is it. Okay, so here we go. Glial cells, first one, astrocyte. Whoops. Here it is in a chart. Talks about the 4 different types in the CNS, the 2 in the PNS, and gives it a characteristic. Okay, so it's summarized there. Here we go. Here we go. Astrocyte, most abundant. Do you think? Duh. Okay, astrocytes are the most abundant. They help with the blood-brain barrier. Now, let me explain what a blood-brain barrier is. When your heart pumps blood up to your brain, blood contains a lot of trash. Since your brain is almost entirely made up of neurons and neuroglial cells, you can't afford to damage any of those neurons. So you have to filter out the trash. The filter is called a blood-brain barrier, okay? And it has several components to it. One is called a choroid plexus, which I'm going to show you in just a minute, a choroid plexus, and the other one is astrocytes and epidermal cells. They act like a fine filter. I'm going to take you back to blood, trying to give you an association here. Remember when we did a blood clot and we said you start off with prothrombin, factor X makes it become thrombin. Thrombin causes fibrinogen to become fibers, and those fibrin, and the fibrin is the long proteins. And we saw it on the blood clot model. We saw those long wires, those blue wires. Okay, those represented fibrin. And in a blood-brain barrier, you have a fibrin type, it's not called fibrin, but a fibrin type of protein that makes a net. And it still is not fine enough. So then what you do is you line that net with astrocytes and epidermal cells, mainly epidermal cells, and that causes a very, very fine filter. Okay, get it? Okay, here we go. I'm going to show you more about that. Let's talk about astrocytes. Okay, so you know it's part of the blood-brain barrier. I made it blue, and it's the most abundant. There's your key. Okay, don't worry about all that stuff. Next one. Oh no, picture dumb. Don't even worry about it. Epidermal cells. Okay, this is going to be blood-brain barrier also, and it's the one, well, I'm going to show you. Okay, now going back to the blood-brain barrier. When blood comes up, it goes through this filter, and then it's captured in a storage area. Storage area is called a ventricle, and we have 4 of them. The storage area for this filtered blood is called ventricles, and we have 4 of them. Two of them are lateral. One is the third, and one is the fourth. So really what it should have been is 1, 2, 3, 4. Yes, wait, let's try that again. 1, 2, 3, 4. But they made 2 of them lateral, and then third and fourth. Got it? That makes 4. And that's where this fluid, which is refined, very filtered blood, is stored and then is distributed around the central nervous system. The fluid is called CSF. Okay, it says in conjunction with other glial cells, the epidermal cells produce cerebral spinal fluid, CSF, and it's in the cord plexus, what I just told you. Okay, cord plexus is the fine, I mean, is the like fibrin, it's the big protein netting, and then we line that with mainly epidermal cells, but also the astrocytes, and the result of filtered blood is CSF, cerebral spinal fluid. Okay, here's a picture. See how fine that is? So any blood would have to be filtered through here. So there you go, CSF. Then with the third type is microglial cells. Uh-oh, look at that one, motile. Motile, mobile. Motile means it's moving, mobile means it has the ability to move. I think it's something like that. Anyway, they have the ability to move. Okay, that's the key to microglial cells. They're phagocytic. So that kind of tells you they move around like a Pac-Man and eat debris to keep the neurons safe. Picture doesn't really show you anything. Oligodendrocytes. Okay, long name. Okay, oligodendrocytes, long name. Okay, characteristic, produce the myelin sheath. Okay, this associated with the CNS only, obviously, is because we're talking about the 4 of the CNS, but you'll see why that is brought up there for you. Okay, that's brought up there because it makes myelin sheath, and it's got a cousin in the peripheral nervous system that makes the myelin sheath also. Okay, they're talking about the way they make the myelin is they wrap around the axon like a roll of tape, produces the myelin. This is the trick. This is the big one. I even put it in red for you. You know I'm going to ask this. Picture oligodendrocyte. Okay, here's the myelin. And what it does, it'll just look like it's clamped around there. But if you looked at it, it's layers and layers and layers of thin myelin, fat. Okay, now we're going to shift into the PNS. There are 2 of them. Satellite cells and neurolimocytes. Long name. Guess what? It's the cousin to oligodendrocytes. And neurolimocytes has 2 names. 2 names. Next to it, you can put Schwann cells. SCHWANN. 2 N's. Schwann cells. I'll show you. I'm going to have it coming up. Satellite cells. They surround the ganglia. Okay, you're saying, what's a ganglia? remember whenever we talk about a new topic, a new system in the body has its own little lingo, its own little vocabulary. And we did this with muscles. It was myo and sarco. Do you remember that? And when we deal with the nervous system, the prefix is always neuro something. Okay. Now, within that category of neurons, of the nervous system, we have other terminology that applies only to the nervous system. If we have a cell body, it's called a cell body. If we have a cluster of cell bodies, has two names. It's called a nuclei or nucleus or a ganglia. If it's in the peripheral nervous system, it's called a ganglia. If it's in the central nervous system, it's called a nucleus. Got it? If it is a huge nucleus, it's called a center. Got it? You'll see what I mean. I got it in a table for you. But you got to learn these terms, because otherwise it'll twist your head. So when you see ganglia, think cell bodies, peripheral nervous system. Okay, now, here is dorsal root ganglia. So we know this is the sensory coming in. Do you see the unipolar? And it's myelinated. And here's the cell bodies, and they're wrapped to be protected, and they're wrapped by satellite cells. Do you get it? Satellite cells go around the cell bodies to protect it. Neurolemocytes. Remember oligodendrocytes? They made the myelin sheath in the central nervous system. The neurolemocytes make myelin sheath in the peripheral nervous system. Got it? Okay. So it's going to be the cousin. So we're going to see in the peripheral nervous system only, and wraps and myelin sheath. Oh, remember the other name? Schwann cells? There it is. Also called Schwann cells. You know I'm going to ask that some form or another. Associated with the PNS only. Remember the oligodendrocytes? It said CNS only. Wraps it like electrical tape. That's exactly the same as the other one. Oh, there it is. Myelin sheath. Okay. Put down the myelin. Okay. And it says same structure and function as oligodendrocyte, except for it's found in a different branch of the nervous system. Good? Neurolemocytes, picture. Boy, we've seen that picture a lot. That's what the model is made out of, made to represent. Here's your axon. This is the myelin. Okay, there's the nucleus. There's the nodes of Ranvier, so on and so forth. There, right there is the axon hillock. Okay. Okay, why have myelin? Do all neurons have a myelin? Nope. Does some neurons have myelin completely surrounding them? Nope, none of them. Myelin, if it's present, is only going to be around the axon. If myelin is present, it's around the axon, it's not continuous, it has gaps. The gaps are called nodes of Ranvier. Got it? Okay. What is the function of myelin? And it's called saltatory conduction. You see it in front of you right now. Well, what does saltatory conduction mean? What it means is instead of being a continuous pluses becoming minuses and pluses and stuff inside, picture the outside of an axon. It's got positive charges. The inside has got negative charges. When an impulse goes through a neuron, the positives go inside and they cancel out the negatives. Got it? The negatives stay inside. They cancel that out. So the inside becomes net positive. Do you understand? And that's how the electricity travels through a neuron. Okay, so it's going to go through at a continuous pace. If we jump from node to node, it goes faster. So saltatory conduction allows the impulse to travel through the neuron faster. This was explained to me this way a couple of years ago, and I thought it was just perfect. On a keyboard, you have the space bar, correct? You have the space bar. If you hit the tab bar, you go 5 spaces at a time. Which one can you move the cursor faster with? There you go. If you hit the tab bar, you're going to go 5 spaces. And that means every time you punch it, you're going 5. You punch it 5 times, you've gone 25 spaces. If you have to hit the space bar, you got to hit it 25 times to go the same distance. It takes you longer. There you go. Okay. So saltatory conduction is to speed the rate of depolarization. Now we're getting into some physiology there, but speeds the rate of depolarization. Okay, it talks about how it's wrapped around there, formed by neurolemmacytes or oligodendrocytes. Okay, we're good on that. I'm going to test you on it, so make sure you got it squared away in your head. And it's an insulator. Okay, when MS, you lose that myelin and it arcs from one to the other, so people's parts of their muscles are jerking as if the brain was telling it to do it, and it's not. Okay, so it's short-circuiting. Good. This shows how you put down the myelin. In your book, it goes on for several pages showing you how you wrap around it. Yeah, big deal. Okay, see how it wraps around it? Okay, you know it's wrapped. Okay, good. What do I want to put here? Nothing. Okay, remember we said that if you lose a cell body because it's injured, your neuron's dead. If you lose the dendrites, the neuron is dead. If you lose an axon, it may or may not be dead. Here's what I mean is, if you're down towards the telodendria, the bottom end of the neuron, you're going to be good. It'll regenerate. The glial cells can repair it. If it's too close to the axon hillock, you're done. So I'm going to show you a picture about that. Okay, it says damaged axons can regenerate if the cell body is intact with a critical amount of neurolemma remains. Okay, that just means there's enough axon there. Regeneration depends on the extent of the damage. Duh. Neurolemmacytes play an important role in that. Well, do you think? Okay, remember the car? Here, your car, let me get the air. Here, your car is damaged. Here, you take it to a mechanic to get it fixed. Okay, if your car is too trashed, you're done. You're walking. Okay, if your neuron is too trashed, you can't move. You're done. Okay. All right. Well, it's called Wallerian regeneration. Here's what it shows is, here's your axon hillock. Let me get your arrow. Here's your axon hillock. You're damaged here. So what happens is this decomposes and then this regrows. Okay. And what it does by regrowing is not, got to remember, this is a cell. A portion of the cell is being repaired, is what it really is. It shows it being regenerated, but it's not. It's being repaired by a glial cell. Okay, you see how it's showing you it's regrowing. Okay, what's doing that is a neurolemmacyte, or not a neurolemmacyte, a glial cell. Got it? We're back to good. Okay, remember when we talked about muscles, and in muscles we had coverings that went around the muscle cell, and it was called mycium. The covering was called mycium, and if it went around the cell, it was called endomycium, and if it went around the fascicle, it was called perimycium, and if it went around the entire muscle, it was called epimycium. Do you remember that? Okay, the neurons are set up same way, parallel again, and and you have a covering, a protective covering. This isn't for sliding. This is just for protection. And if it goes around the neuron itself, it's called endoneurium. You change the suffix from mycium to neurium. So if it goes around the cell, it's called endoneurium. If it goes around the fascicle, and we're going to take clusters of axons before they're a nerve this big. Clusters of axons this big are called fascicles, just like they were in neuron. Got it? So we have a covering that goes around that, and that's called perineurium. And if it goes around the entire neuron, it's called epineurium. There you go. There you go. Look at endoneurium, goes perineurium, fascicles, epineurium, entire nerve. Good? You know, I'm going to ask you on an exam. The prefix is just like it was with muscles. Only thing is we changed it from mycium to neurium. Are we good? Okay, here's a picture. The outside is a nerve. Do you see this covering? Epineurium. So this is an entire nerve made up of these are neurons right here. Okay? These are neurons, axons. So the covering around that is an endoneurium. And those are clustered into fascicles, and the covering around that is called perineurium. Pretty straightforward, huh? Okay, picture, you saw it. This shows that we're not making it up. The only thing they did with this one, which is kind of jacked up, is they took and did it from the end here, and they did it from the side here. So this thing really doesn't have anything to do with this one, okay? Because you can't see the individual bundles. These are fascicles. Okay, there's the axon, this is a fascicle, and the coverings and so forth, okay? And the whole thing, this is a picture of a portion of a neuron. I mean a nerve, okay? Synapses. See who's calling me. Okay. Synapses. We've already gone through this. Okay. The only one is this one is neuron to neuron instead of neuron to muscle. Neuron to muscle was called neuromuscular. This one's called neuro. Okay. Okay. We know, remember the presynaptic membrane, synaptic vesicle, neurotransmitter. Good. Postsynaptic membrane, neuroreceptor. In between is a synaptic cleft. Chemical, the neurotransmitter, which is a chemical, travels across there, opens up gates, allows sodium to go in. You remember the picture? Okay, I did that one on directed teaching and drew it for you. And then the synaptic cleft is the distance between the pre and postsynaptic neuron. Okay, don't look at this one. It doesn't really mean anything. See this one here? Okay, this was in the muscle, this was a sarcolemma, but this was the gates. And the neurotransmitter, which is green, holds open the gate. And I think the red represents sodium or the green represents sodium. One of the two. Anyway, here's the synaptic vesicle. This to show you there's still proteins inside and mitochondria and that kind of stuff. Okay. What breaks the synaptic vesicle is calcium. And there's calcium gates down in the synaptic knob. They allow calcium to come in. They break the synaptic vesicle, releases the neurotransmitter. Okay, yeah, the red, it says acetylcholine, that's a neurotransmitter, is red there. So the green represents sodium. And that's what you want in here. Okay, where's my little arrow? That's what you want in here is the sodium. Okay, remember we talked about converging and diverging. That was it, converging and diverging. I don't remember what the hell I said, but I said converging and I said something else. Anyway, converging and diverging. These are when converging is when many neurons come in to meet one, to bringing in information, okay? Diverging is when one branches out into many, okay? So most of the time we see converging on the sensory side, the afferent side, and we see diverging on the efferent side, the motor side. Don't worry about reverberating, don't worry about parallel. Here's a picture. Do you see, now here, see the input? I mean the output, the input, this is converging. Many neurons leading into one, converging. Here's diverging, okay? One neuron leading into many, that's diverging. Good? Don't worry about the other two. Okay, development of the nervous system. We said we weren't going to do that. Good? Okay, it just shows what kind of tissue it's coming from. Development, yeah? Guess what? Guess what? Done. Now, I'm going to post this one, and then the next one is the brain. It's a long one, okay? So I got to have a rock star. but I'm going to sign you off here and get this one loaded up for you. So let me get down to stop recording. Okay, see you guys.